Good morning to those of you who are logging in now as attendees to our speaker forum, Pediatric Speaker Forum. It is 10:29. We will wait another few minutes for folks to get logged in and comfortable. So don't go anywhere.
Anyway, if your Wi-Fi starts to slow down.
I believe some participants thought we are muted. Todd is, Monique is. I hope they can hear me.
Yeah, good point. Thanks. I was missing a couple things popping up in chat, Dr. Ritz. Thank you for that. Yeah, you're not hearing us yet because we were muted. We were just stalling for another moment or two while folks logged in and got comfortable. So we'll start in about 30 seconds. Okay, let's get started. It is 10:33. I know we've got quite a few attendees on now, and we expect to have more logging in as we walk through an intro here. But good morning and Happy New Year, and welcome to Pediactive's 8th, I believe, virtual speaker forum. We had many more prior to going into the virtual space, but this is our 8th virtual speaker forum. My name is Todd Morrish, and together with Monique Buffler, we will be your hosts and moderators this morning. Monique, say hi from beautiful Park City.
Good morning, everybody.
So before we introduce our speaker, a little bit of housekeeping. First, the Pediactive Board and the Pediactive Disparity Committee welcome your input and involvement in ways that will help increase the diversity of Pediactive and help address the healthcare disparities for people of color with PD. You can help us by connecting us to people you may know at community health centers and other community-based health programs in Alameda County. If you have any expertise in this field of work, please let us know so we can tap into your knowledge. Please send an email to this address to express your interest. It is administrator@pediactive.org, and Monique can paste that into the Zoom chat window for you all, and we'll be using that chat window for other information from us to you rolling forward. Next, we always like to cross-promote our many Pediactive programs. Besides Pediactive's renowned support groups, we also offer many other classes and programs for people with Parkinson's and their caregivers and loved ones. There's Tai Chi and yoga and Pilates and meditation, and there's voice and singing and boxing and dance and much more. If you have tried one or more of these, we ask you to please tag a friend or family member next time to join on your next session. Please check out the calendar link on pediactive.org and we'll paste that URL to the calendar into the Zoom chat tool now as well. So as you may have heard when you logged in, this webinar is being recorded, and we will post that recording to our PD Active YouTube channel in the next few days. Again, see the chat window, and we'll put that URL there as well. And by the way, all these URLs and all these pieces of tidbits of information that we're putting into the chat, we will probably also be sending out in a follow-up to all attendees after the fact as well. So if you can't scramble fast enough to copy and paste out of chat, don't worry about it. It'll be coming into your inbox soon. You may submit questions when you have them using Zoom's Q&A feature. They're in your Zoom tool tray on the bottom of your screen. We have reserved Q&A time at the end of the presentation to pose those questions to Dr. Ritz. Monique and I will use the Zoom chat feature, as mentioned, to relay information to you regarding the presentation. But we do prefer you guys use the Q&A tool for questions for Dr. Ritz. Throughout the doctor's presentation, we will present you with a handful of poll questions, which will pop up on your Zoom screens. Please answer these as quickly as you can so Dr. Ritz may respond to those in the flow of her presentation. If you prefer to not answer the questions, merely close the windows when they pop up. And we will start with 2 quick poll questions right now to get into the swing of it. Here's the first one. So we'll let that run for a minute just so that Dr. Ritz can see who her audience is today. And then I'll pop up a second one here in a moment. If for any reason Dr. Ritz or I or Monique get disconnected from this webinar, which I highly doubt, please be patient and do not disconnect or leave the meeting for a few minutes as we try to get reconnected. It is possible that if I get disconnected, Monique may take over as host if I can't jump back in, but I doubt that will happen. Hasn't happened yet. Finally, PD Active depends on donations to bring events like this to our members free of charge, and we remain super grateful for your contributions. If you had not yet had a chance to make the suggested registration donation of $10 for the speaker forum, we will paste that link into the chat tool now. So let's end that particular quick intro poll. So there you see Dr. Ritz, as predicted, most of our members are actual PWPs, or people with Parkinson's. A fair number of caregivers and those that work in the community and family members. So let me jump to the next one, because this is oops. Here you go, folks. Have you read this book yet? Ending Parkinson's Disease, A Prescription for Action by Dorsey Scherer Oakenbloom. So we've included this question because, one, it is an excellent layman's look into not only the causes of PD, but also what we as individual lay people can do to help prevent it. And 2, much of it overlaps with our topic today. For those of you who have not yet read it, we strongly encourage you to do so. If you cannot afford to buy the book, then email them at info@endingpd.org, and they will send you a free one, I understand. Is that right, Monique? That's cool. Did not know that. Okay, I'm going to end and share the results of this poll. So some 20% of our attendees have, and some 28 have not yet, but plan to, and another 50% haven't. So like I say, it's a great read. Highly recommend it. In fact, at the end of today's forum, Monique and I will review with you a brief summary of action steps we can all take in the fight against PD. Okay, here we go. Dr. Beate Ritz, let me close this down. Dr. Beate Ritz joined the faculty of the School of Public Health at UCLA in 1995 and is professor of epidemiology with co-appointments in the Environmental Health Department at the UCLA School of Public Health and in Neurology, UCLA School of Medicine. She is a member of the Center for Occupational and Environmental Health, the Southern California Environmental Health Science Center, co-directed the NIEHS-funded UCLA Center for GeneEnvironment Studies of Parkinson's Disease, and is interim director for the APDA Center of Excellence in Parkinson's Disease Research. Dr. Ritz's research focuses on the health effects of occupational and environmental toxins, such as pesticides, ionizing radiation, and air pollution on chronic diseases, including neurodegenerative and neurodevelopmental disorders like Parkinson's disease. Since 1997, Dr. Ritz has been investigating the long-term effects of pesticides, pesticide exposures on Parkinson's disease and cancers, and is currently conducting a project to implement Parkinson's disease registry as required by a 2004 law in California. In 2011, she received an award for the American Parkinson's Disease Association for outstanding contributions to the advancement of Parkinson's research. Wow, Dr. Ritz, your background is extremely impressive, and that is probably a subset of all that you've been involved in. I'm sure I speak for all of us in thanking you very much for all you do to help our cause, and welcome and thank you also for taking time out every weekend to speak to our community today. I'll hand the figurative mic over to you now, so take it away, please.
Yes, and I will share my screen. Oops. Here we go. Sorry. Okay, I hope you can see this. And
Yes.
I'm always having a hard time with the seeing the slides versus me. So I'm going to put this in the corner. Well, first of all, thank you very much, Todd and Monique, for inviting me and giving me the opportunity to actually speak about something that I'm extremely passionate about and have been doggedly studying for almost 25 years. In fact, it says Todd told you that I've been studying it since 1997. My interest in this area was, however, already there when I took the job at UCLA. And I looked around the state of California and I saw two environmental issues. One was air pollution. In the 1990s, it was a huge issue for California. It has become better, but it's still not solved. And the other one was, of course, in the rural communities, the issue of using pesticides at an industrial scale. And unfortunately, while I was able to study Parkinson's and neurodevelopmental disorders, autism and others as well, with respect to this exposure, it has only increased. So we're using now more pesticides than ever. And when I started, California already was using 25% of all the pesticides applied in all of the US. And part of that is not only that it's industrial farming, it's also that we have multiple growing seasons, different from other parts of the country. We can actually really grow crops more or less all year, and pesticides are helping us do that. And they have a function and a worth, of course, for the industry and for our food supply, but they also are problematic. And why they are problematic, I want to share with you today. So I'm going to talk about environment and Parkinson's disease. I already said I'm going to talk about pesticides. And the main study results that I will refer to and talk about are the PEG study. And that is that study that I got funded in the year 2000 and that I just got refunded for the third time. That also contributed to the NIEHS Center for Neurodegeneration with its human research, and is basically responsible for most of the results I'm going to share with you. And another study on air pollution that I actually conducted in Denmark, and you see here a Danish icon from Copenhagen, and that's called the Pesticide study. And in Denmark, I studied air pollution, and I'll share some results with you. And then more recently, we also ventured out and looked at metal exposures, primarily lead. And we did that again in the PEG study, but we also used samples from colleagues in Australia. And a colleague of mine in Harvard has been doing a study as well. What I will not touch today is trichloroethylene. That's a subject matter that Dr. Tanner and her colleagues dug into a while ago in workers who are using this solvent. It's a solvent, and there's some data out there, but I myself have not studied it, so I'm not going to talk about that today. But we have enough to do.
And Dr. Ritz, PEG stands for what?
PEG is the Parkinson's Environment and Gene Study, and it will be spelled out when I get to it. So, great. So what about pesticides? Well, you can see here that, pesticides were widely used, or chemical pesticides, industrially produced pesticides, were widely used more or less in the 50s, or the use started, and it has only gone up. The production, as well as the imports and exports of pesticides. The earliest one was DDT, and Muller, who invented DDT in 1937, got the Nobel Prize for it because it really helped curb typhoid in World War II and saved a lot of lives that way. And then in the 50s, it was used widely on crops to kill insects. We can also see that, while the US is number 2, I don't know whether you can see that, is number 2 worldwide, China leads in the production and the consumption of pesticides nowadays. And that's, China is also a big agricultural place, but you see that Argentina is just behind Thailand, Brazil, Italy, France, Canada, Japan, India. India, relatively few, given that it's such a large subcontinent. But overall, it's millions of kilograms of pesticides applied every single year in an industrial manner. In California, we have something called the Pesticide Use Reporting System that allows us to actually map the pesticide use. This system was created in 1972 by law and then instituted in 74 and is running. And here you just see the paraquat use on commercial fields in the Central Valley, and you see it also has a little bit of use in other places, but mostly in the Central Valley, which is the place where my study is actually happening. And here you can see that in the US between 1990 and 2007, most use was herbicides in terms of tonnage. And paraquat is actually the most widely used herbicide. For a while it was outnumbered by glyphosate, but now it's actually paraquat is coming back because a lot of weeds are getting to be resistant to glyphosate. And then the second row here is insecticides, and those I'm very concerned about as well. I'm concerned about paraquat, but I'm also concerned about insecticides. And the reason why I'm concerned about insecticides is because they are intentionally neurotoxic. So they are engineered to kill. And they kill and the way they kill is by poisoning the nervous system. And of course, you can say, well, insects are tiny and they may get their nervous systems wiped out. But what about humans? We are certainly not dying from it. No, we don't. But there might be long-term chronic effects on our nervous system, and maybe neurodegeneration is one of them. And that's what I was studying. Then the yellow one, you can't read, it's fungicides and bactericides. Fungicides are highly toxic, and they are also toxic to nerve cells in special ways. And bactericides, I'm now really interested in because we recently learned, and I will talk about that a tiny bit here, that actually the bacterium in the gut may be quite important for Parkinson's disease, for its onset, but possibly also for its progression. So why should we worry about pesticides if we're not farmers? Well, we should. The reason is actually given by some surveys that the National Institute of Health pays for, or the government pays for. They're called NHANES, the National Health and Nutrition Exam Studies. And these are done every 4 or 5 years. They are representative of the US population. And you will find that a subpopulation of the people who answer questions about their health also give urine and blood samples. And the CDC lab is actually examining these urine samples for all sorts of, and blood samples for all sorts of environmental pollutants, including urinary pesticide metabolites. And you can see here that in the year 2000, when my study actually started, 96% of all samples were positive for chlorpyrifos, we will encounter chlorpyrifos in one of my slides, and I'll remind you of this. And about 30% of all samples were positive for diazinon. So almost all of the US population was exposed in the ages 6 to 59 to chlorpyrifos in one way or another. Why? Because we used it indoors and in the garden to kill insects. So it was widely used indoors. That's not the case anymore because it was banned from indoor use in 2004 because of science, and it was actually children's science, neurodevelopment that made that possible, and it's also banned in California. However, it's not banned for agricultural use. It's only banned for indoor and garden use. Diazinon is also one of the most widely used agricultural pesticides, and both of them can be found in the air around communities that apply these professionally. So this was in the year 2000. The use of these pesticides has gone down. However, now we are worried about other kinds. One of them, the so-called neonicotinoids that were introduced about a decade ago to replace these. And these are insecticides that are now being found in half of the US population's urine samples in NHANES. So even so, they're mostly used in agriculture. They also have home uses now, and we again are exposing just about everyone. All right, that said, why pesticides and Parkinson's? It's actually kind of an old story it was an old story when I started this study. That's why NIH funded it, because they knew there was something wrong out there. That actually was linked to Parkinson's. And it's all of these studies that are summarized here in 2006 by Brown et al. This is called a meta-analysis, and all the dots on the right of this line say there's an increased risk. And what is the increased risk? It's the increased risk of having Parkinson's if you report having used pesticides or specific pesticides in your lifetime. And these odds ratios, these are ratios of exposed over unexposed. And if they are greater than 1, it means the exposed have a higher risk. And they have a here 90% higher risk. So 1.9 gives you a 90% higher risk, almost a twofold increase in risk with pesticide exposure. These studies also found that rural living increased your risk of Parkinson's by 60% and farming by 40%. Okay, we know it's pesticides, it's rural living and farming, and probably rural living is usually healthier. It might be the pesticide exposure and not something else. But which pesticides? Is it important that we know which pesticides? I think yes. Okay. So that is why actually the NIH decided in the year 2000 to give me a grant that's called the Parkinson's Environment and Genes Study Grant, or PEG, and that's the study that recently was refunded 2 years ago with a new hypothesis that I will show you later. In the meantime, over the last 20 years, this study collected almost 850 Parkinson's patients who we saw clinically and also followed over time. So we have not only onset data, but also follow-up data for these individuals. And they all live in 3 rural counties, Kern, Fresno, and Tulare counties. You see them here on the California map. And from these communities, we also enrolled about 1,000 population controls. And I just have to thank everyone who participated, because these people went through days of data collection with us and also gave us blood, saliva, urine, and more recently, fecal samples for us to do investigations on. And the person I also want to thank from my collaborators is Dr. Bronstein, who has seen most of these patients. So we drive up from LA 4 hours in a car to Fresno, set up clinic, and see these individuals personally. Now with COVID, we are doing Zoom meetings, and that works as well. Okay. Sorry. So what do these people do? Well, they give us a lot of data. This is part of the data they give us. They give us addresses. They tell us whether they lived on a farm, what their water supply was, whether they lived near fields or towns, and also do the same thing for workplace addresses. However, that would have not given me the grant. What gave me the grant is the California foresight to have this pesticide use report system in place. And what does this system do? It provides us with data from everybody who's a farmer or applies agricultural pesticides, where they apply it, especially within a 1 square mile area, when they apply it, on what they apply it, what method they are using, how many acres, how many gallons, and what the active ingredient is of a specific pesticide in pounds. So with all of that information, I and my students generated a computerized system that you see some printouts here. This was a paper map that we computerized that then looks like this. And then we used all of this data and overlaid it with the maps. And you can see these maps just give us landscape views of what the crops are. And here you see these are orchards, these are vineyards, these are field crops. And then we overlay that with the information from the PUR system. And you can see, we now can see what the pesticide was applied on in this year, 1988, around the tiny community of Shafter, for example. And these are homes, and you can see that these homes were not close to any application of pesticides, but look at these homes. They were really close, right? And why is that important? Well, because we now can plug onto our maps the address for every single individual in our study. At the time they lived at this address, we can draw these circles around the homes, and we can say how much of a certain pesticide was applied within that circle. And that's what we did for every single person. After we plugged their address in, here you see a Google Maps image of it, where we tell you how we find their addresses and where we know and how we know where they lived. And we not only did that for where they lived, we also did that for where they worked. So anybody who, for example, may have worked at a school like this would also have this way for us to draw the circle and determine what was applied while they were working. And if they worked on a farm, this is an aerial photo, we would know what was sprayed on the farm around. It's a fantastic system that we used, and that, you know, allows us to map these uses of certain pesticides over the time. And you would say, well, this might have been applied, but has it really, you know, affected people in the homes or in their workplaces? This is what EPA did. Pamela Woolford and her colleagues put monitors, air monitors, on the rooftop of primary schools. This is the little town of Parlier. These little flag thingies, I don't know whether you can see them, but they're these little ships with flags. These are monitoring equipment on the rooftop of primary schools. And what they did is measure about 30 pesticides in the air on the rooftop of primary schools in these communities, and then also use the PUR, the pesticide use report system. And here you see the mapping of what they did to know whether certain pesticides were applied around the area of this little town. And then this is the data they generated. You see the weekly average chlorpyrifos detected in the air with the monitors. So that's these bars, right? And then the weekly sum of chlorpyrifos applied are these kind of pinkish dots. And you see that, you know, farmers applied, and you see it was monitored on the rooftop of schools. And then they didn't even apply, but there were some residues that were still monitored, and then they applied again, etc. So you can see that the application pattern around these areas where they monitored very nicely coincide with what was monitored in the air. And no wonder, right, that we could measure chlorpyrifos in the year 1999 in 96% of all urine samples of a representative cohort of US citizens. So that was chlorpyrifos, but the California story actually started with what I call a very unfortunate natural experiment. And this unfortunate natural experiment was actually a designer heroin contaminant called MPTP. And this MPTP, look at the structure, the chemical structure of it, looked very much like paraquat. And that was the herbicide I talked about before. It lacks, MPTP lacks this group on this side, but industry was already on its way to actually generate exactly this molecule also as another herbicide. When it was found that, you know, MPP plus, it's actually this agent that's active with the N plus here, is killing dopamine neurons. Here we have dopamine, and this is another pesticide that actually interacts with paraquat nicely to give animals in animal models Parkinson's-like features. So what happened in this natural human experiment? Well, somebody in their garage tried to evade prosecution for making heroin by making this designer heroin, and in the cooking process, something happened that generated MPTP, and the injection drug users of this batch of heroin actually developed within a few weeks instantaneous Parkinson's disease. And that's very well described in the Frozen Addict book by Dr. Bill Langston, who investigated this outbreak, this cluster of people developing Parkinson's instantaneously in their 20s and 30s and 40s, something nobody had seen before. And that got us into investigating herbicides, specifically paraquat at the time. So with my PEG study, one of the things I did is I looked at the residential addresses and the occupational addresses. And indeed, if both paraquat and MANEP, which was the one that also in the animal models promoted the death of the dopamine neuron when it was given together with paraquat, both of them increased the risk of, especially in occupational settings, but also in residential settings, about 80% the risk of Parkinson's in my PEG patients. We also, so this was presuming that you lived close enough to a field where some farmer had applied this paraquat to the field, and you know it whiffed over onto your property, and you had breezed it, or you were in the vicinity doing whatever you do occupationally, and you got a good whiff of paraquat, or it stuck to the soil and you brought it into your homes. Another study we did was well water consumption, because about 80% of the people actually in these counties were using well water, and the wells sometimes were not very deep. And we decided to ask them about well water use and to see what was applied around these wells. And we found that if there were water-soluble pesticides applied around the wells of these individuals, you had about a 68% increase in risk. If it was organophosphates, which is chlorpyrifos, our favorite, you had about a 70%. If it was a carbamate, it was a little bit less, but all pesticides, we had about 26 of them, gave you about a 66% risk increase if you were drinking well water, and especially if you were drinking well water for a long time. The next thing we did is we wanted to see whether people are at different risk because they are more susceptible to the neurotoxic effects of these pesticides. And we call that a gene-environment interaction in this case, because the genetics, as Dr. Bill Langston always said at these conferences that I went to and talked to him, genetics loads the gun for the disease, and then the environment pulls the trigger. So basically we need something to load the gun, so we have a susceptibility to come down with a disease, and then the environment acts on top of it to actually give us to pull the trigger and make the disease happen. If the gun was never loaded, we wouldn't have the risk, but also if the trigger wasn't pulled, we wouldn't have it either. So that's how we can imagine this gene-environment interaction works. So what do we do with this knowledge? Well, we can actually look for people who are at risk genetically. And this was a colleague of mine, Dr. Kilada, together with Dr. Checkaway, who I've been talking a lot. He's doing a lot of occupational studies for Parkinson's. And they identified a dopamine transporter gene that put you at risk. And they said, what happens if you then are also pesticide exposed? And they did this study in Washington, which has a lot of fruit and vegetable farming as well. And they looked at who had more of one risk allele. So you can have two alleles on your chromosomes. And if you count these alleles, then you could have one risk allele for this dopamine transporter, or you could have two risk alleles for the dopamine transporter. But you see that the risk for Parkinson's increases very little with the genetics. It increases a slight bit, but not much. But when you're then hit by the environmental agent, in this case, pesticides, that's when your risk increases, right? And as much, if you have two of those alleles, two of these genetic factors, then it's almost fivefold, or in their case, more than fivefold. At the time, I didn't believe them. I thought this was just a statistical fluke, nothing you could ever reproduce. But Dr. Checkaway kept bugging me. Beate, you have data, you should look at this. And I finally did, and it was published in 2009, so 3 years later. And look what I found. We counted the risk alleles in our subjects and controls, and we also looked at who was paraquat and monob exposed, just like we saw before. And you can see here, yes, the risk alleles put you at risk a little bit, increase in risk here, especially when you have two of these alleles. But what really pulled the trigger was the environment. Being exposed to monob and paraquat increased your risk with one allele 2.5-fold, and with two almost fivefold, almost exactly the same the way they saw it in Washington. So this is a replication of a previous result we were able to do. So we also looked at environment interactions, in this case head trauma, because it has been hypothesized that head traumas increase the risk of Parkinson's. And we had in our interviews asked patients to report on head traumas that they had in their past. And not just head traumas, but head traumas that actually knocked them out and knocked them unconscious for about 5 minutes minimum. So we call that traumatic brain injury. And we then looked at the risk of developing Parkinson's. No brain injury and paraquat, you had a slight increase in risk. Having a traumatic brain injury and no paraquat exposure increased your risk also. And in this case, it was actually a stronger risk factor for Parkinson's. And you can now put these two stacks together. Here is the risk for paraquat alone. Here's the risk for head trauma alone. But these are the poor individuals who had both. Threefold risk increase for Parkinson's if you had a head injury that knocked you out and unconscious, and you were paraquat exposed. Okay, so again, not everybody exposed will get the disease. But if you add things together, finally that bucket will overflow, or that, that bucket is full and it overflows. You don't have enough neurons left in the end. And we call that an environment interaction. Now I'm coming back to my pesticides, my favorite pesticides I talked about before, the insecticides that are actually known as active neurotoxins. In fact, all organophosphate pesticides have been developed from sarin gas, which is the nerve agent that was used in World War I to poison soldiers of the other army, hopefully. But it was not a good idea for anyone. And these organophosphate pesticides poison the nervous system. The way they do it is by being metabolized in the body and shutting down acetylcholine esterase. And so there is metabolism involved, meaning our metabolism first activates this pesticides to the actual toxic agent. And then there are enzymes that detoxify so we can excrete the agent and get rid of it. And the good news is that we actually get rid of these kind of pesticides really fast from our body. So within 48 hours, they're in, do their damage, and they're out. In a way, you can say at least we get rid of them, right? On the other hand, it's really hard to study them because I cannot go and take a blood sample and find them unless you have been recently exposed. So you can only find them is that if there's chronic long-term exposure, somebody who was harmed 40 years ago, 20 years ago, now has Parkinson's, I won't find it, okay? At least not the active metabolite in their blood. Again, why am I interested in gene-environment interaction? Because we know that the one enzyme that detoxifies and gets rid of this active ingredient that kills neurons is actually paraoxonase. It's an enzyme that is known to be genetically very different across populations in its activity, in its ability to detoxify. So we have populations that are, or people in a population that are so-called poor metabolizers just because of their pure luck genetically. And we have people who we would call ultra rapid metabolizers. So these are the guys who get rid of the toxic agent really fast. And how do we use this in my study? Well, we can actually determine who is a poor metabolizer and who is an ultra rapid metabolizer by looking at two genetic polymorphisms. They are called variations, genetic variations. One is called the 55MM and the other one is called the 192RR. Putting them together, or QQ, you see that this 6 is much lower than this 22, right? And that's the rate at which this enzyme is active. It is active at eliminating 6 microgram of whatever, you know, this organophosphate is per minute versus 22. So this is clearly almost fourfold faster than this person, right, in eliminating. So knowing this, we can now classify the population we just studied into poor and high metabolizers. So these are the guys who can metabolize organophosphates really fast. These are the guys who are slow, just genetically slow in their enzyme activity to detoxify. Now we have diazinon, chlorpyrifos, and I also gave you parathion because that's another common pesticide used, of the OP classes. All are being detoxified by this PON1 enzyme. If you have 0 exposure, PON1 does not increase your Parkinson's risk very much at all. However, if you have exposure to diazinon, to chlorpyrifos, to parathion, and you are a high metabolizer, well, there's a tiny bit of increased risk for chlorpyrifos, but it seems like it doesn't increase your risk for parathion or diazinon at all. But if you're now a medium or a low or slow metabolizer, look at how it steps up to a 2.5-fold Parkinson's risk, a 3-fold Parkinson's risk, and again, a 2.5-fold. Okay, so you have to have both. You have to have the inability of getting rid of a toxin and you have to be exposed. That's why you might get sick and your neighbor doesn't, because your neighbor might have the very fast enzyme. So this result that I just showed you was based on the POR system, that pesticide use report system, people living in the community being bystander exposed. We also ask everybody to report on their household pesticide use. And then a student spent about a year to classify the names of the pesticides we were given by everybody into these different kinds of pesticides that were used in the home. I already told you that chlorpyrifos was indeed a pesticide that was very much in all of these insect repellents and insect anti-insecticide agents in the 1990s and in 1980s as well. And we went to all these databases to find out which of the raids and wasp hornet, whatever toxins actually had active organophosphates in them. And now you can see what happens when we are asking people, do you use any household pesticides and which ones? And then we classify them into OPs or not OPs. So the never users or rare users of any pesticides have no increased risk in this is the reference group, and the ones that are using pesticides that didn't have OPs had a very slight increase, but it wasn't statistically significant. And then we have the frequent users of the organophosphate pesticides, and they also didn't have a risk increase at all. It's 1, and this confidence interval tells you it's not statistically significant. But look at what happens when we now have the slow metabolizing individuals that cannot break down this organophosphate, a 2.6-fold risk increase. If you are a slow metabolizer and you used in your household one of these that actually had an organophosphate in it. So basically my study is telling you something. Well, it's not good to live near fields where there's application, but it's also not good to use them in your household. So two totally different exposure assessment methods give you the same result in this case. In addition, we actually, In addition, we also not only looked at the onset of Parkinson's, but I told you before we had followed patients over time, and most of you know and fear that your cognition gets worse. And yes, the cognition did become worse of some of our patients, not all of them, but some of them, over the time that we followed them. You can see here, however, that was also differential by exposure. Those who had high exposure to these organophosphates and were slow metabolizers lost a lot more cognitive points, abilities in this case, than the ones who were not exposed and were normal metabolizers. Okay, so we see here again that even when you have Parkinson's, these pesticides are definitely not good for you in terms of progressing to some of these outcomes that you don't want to progress to, like cognitive decline. We played this game of gene-environment interactions with lots of different pesticides, lots of different genes. Our colleagues took these into the lab. They did some mouse studies, they did fly studies, they did fish studies, they did cell studies. And there are tons of publications you can find that we contributed to with this kind of approach. Something else we started working on was looking at the DNA and the genetics in a different way, where we are closer to the environment. And that's what we call epigenetics, where actually the environment does something to your genes. And what does the environment do to your genes? It tells them whether to produce a messenger that then produces proteins. And the environment can influence this whole message translation signaling. And that makes sense because, you know, blood cells that encounter a lot of of this toxic agent, organophosphates, who are inhibiting acetylcholine esterase, these blood cells may want to make more acetylcholine esterase or may want to turn down their receptors. So there's a back and forth in biology between, you know, what you put in and what the cell wants to do with it. And basically this back and forth is what we used here. And it's called epigenetics, and we're looking at epigenetic marks that the environment leaves on the genes. And why is this important? Well, first of all, we found that the environment interacts quite a bit with the genetics of blood cells and also the composition of the white blood cells. For example, we find a lot more granulocytes, which is the most common type of white blood cell that actually controls bacteria in your respiratory tract as well as in your gut. So this leads us to think, well, if Parkinson's patients have more granulocytes, do they have to defend themselves more against infections, for example? Also, what we used this epigenetic readout for is to actually look at what kind of signals it leaves in terms of pesticides. And this is an incredibly talented young colleague of mine who found organophosphate signals in the blood after long-term exposure. And why am I so excited about it? Because I told you that these pesticides are like, ninjas. They go in and out in 48 hours and, they leave damage, but they don't leave a trace of who they were. Well, we now actually have a trace because cells were fighting them, and they were fighting them in a very specific manner. And that left kind of a signature for this agent. And that's what she was able to show in this beautiful paper she wrote. So we found these long-term organophosphate signals in the blood of our exposed individuals.
And her name, Dr. Ritz?
Dr. Kimberly Paul.
Kimberly Paul.
And sorry, I should have put her name on the slide. So what are we doing with all of this now? We are actually going really out all the way for gene-environment interactions with colleagues at Harvard, Dr. Vic Khurana and his group, and they have raised cells in a dish. And these cells are actually human cells that contain our favorite protein, alpha-synuclein, that's responsible for the Lewy body. And these cells were from Parkinson's patients and are now used in toxicologic experiments where they put these different pesticides into the lab dish to see what happens to these cells. And this is a study that is funded by the Department of Defense and is unfortunately running out of money at the end of this year. But we have been able to actually show that what we saw in our human subjects as increasing risk of Parkinson's, they could reproduce in the lab with human cells that they turned into dopamine cells. We also are now going after what has been called a changing face of Parkinson's, like does it really start in the brain or does it start in the periphery? Which periphery is it that I'm talking about? Gastrointestinal problems. Why? Because the gastrointestinal system actually is known to interact with the brain. And we now think that some Parkinson's disease may start in the gut, be promoted in the gut, and that gut bacteria contribute to it, may promote what is going on, may metabolize some of the toxic agents, and that's what I'm interested in, or that the toxic agents actually are responsible for the composition of the bacteria in your gut and the metabolites they generate that then go into your bloodstream or go up your nervous system into the brain. And that is the latest hypothesis we are now following, and that's why we need fecal samples to look at the gut microbiome of our patients. And we already have found some differences in the microbiome of Parkinson's patients and controls, but I'm not going to talk about this much because the paper hasn't come out yet. It's the doctoral thesis of one of my students. We are also looking at metabolites because that's what we think is actually responsible for some of the toxicity. And Dr. Kim Paul is actually generating these beautiful images of different metabolites she can find that are actually different between Parkinson's patients and control subjects, and that might actually tell us a lot more about what the toxins are and also what Parkinson's patients are encountering compared to control patients. So these are metabolites of Parkinson's patients that are in red that have gone up. These are metabolites that have gone down, and we are trying to decipher which of them are toxic or are maybe a defense mechanism in Parkinson's. Why is all this research important? Well, it's important for standard setting for neurotoxins, and it's also important to reduce the risk for Parkinson's disease, and we need to know more about it. We also need to go towards having something called a pesticide vigilance system, which the name is taken from pharmacology or pharmaceuticals, where we have a pharmacovigilance movement. That is a system where after the introduction of a medication in the medical practice of a country, the pharmaceutical industry is actually held responsible for what the side effects are in the population. So they have to spend a lot of money on so-called phase 4 monitoring, which is a monitoring system of what is happening after people who take the drugs for good reasons. What are the side effects and what should we learn from those side effects in populations? And what this kind of principle of pesticide vigilance says, yes, if we need pesticides, at least the industry should then go out and monitor and protect human health after they have introduced it widely. And that does not happen currently. So this is a perspectives article from Science magazine that if you're interested, I can share it. I also want to briefly cite E.O. Wilson, the big ecologist who died this year. He was a Harvard colleague who studied insects his whole life. And he said, if insects were to vanish, the environment would collapse into chaos. And the reason why he said that is because insects support terrestrial food chains, serve as the plant's chief pollinators and crucial decomposers. So while we want to save our food supplies from pests, we call them pests, in the end, they are insects. In the end, we also need them for food supply and for pollinating. So I think it's time to rethink how we actually deal in agriculture with these kind of, vast applications. So I stop here for a second. I have some slides on air pollution and on lead, but I talked for a very long time. Do you want me to keep going or just say that, this is for another time?
I think I'd like you to keep going. You've got some incredible data there around the demography of the rural populations and farming communities, and a little bit in terms of home products for non-rural environments. But I think for us immediately in these urban areas that we live in here, air pollution is a concern. If it isn't, it should be. So we'd like to learn a little bit more about that, and then we'll lob you a bunch of questions after that.
Okay, great. Yeah, so definitely in rural environments, that's what I studied. The reason why I was studying it wasn't because that's where the only risk is. We are also exposed to pesticides in our homes, but also through our food. And it's much harder to study food and contamination with pesticides than it is studying pesticides the way I did in these rural communities. But the principle applies to everyone who's exposed. So it doesn't matter whether it's through the air, through the water, through your food, or through home use. But now let's go to air pollution. So air pollution has a long history, and this is just to show you that it started in England in the 13th century with big laws against, burning so-called sea coal. And then again, England in 1956 was the big London fog episode that restricted coal burning to and created the UK Clean Air Act and eventually also the US Clean Air Act in the 1970s. A similar incident in Donora, Pennsylvania, where people actually died from air pollution, but many more fell sick.
And it was interesting that it was in those window of the 1950s that Dr. Parkinson actually started noticing people walking around with early stage symptoms of Parkinson's in the big fog.
That was 1830.
18, right? Thank you.
Yes, it was that was during the Industrial Revolution in England. Exactly, that's when he saw them walking around the way they did, and air pollution was certainly really bad in London at that time. But it did get better over the 100 years, but really public health interest only was sparked in 1956 by this big episode where you could actually see how, smoke increased and death increased. So this was the smoke and this was the number of deaths. And it kind of stared you in the face. So the global burden of disease in 2010, so more than 10 years ago, already identified air pollution, specifically particulate matter air pollution, as one of the 10 most common risk factors due to which healthy life years were lost and deaths were increased. In 2019, they re-ranked, and now air pollution was actually number 4. So it has gone up from number 9 to number 4 worldwide. Of course, partially this is due to the increasing air pollution in low and middle income countries, including India and China. But it's definitely out there and something to take into account. What I did, I actually went to a relatively low air pollution country, Denmark, quite clean. It's an island nation in the ocean. And in this place in Denmark, I had a chance to do an air pollution study for multiple reasons I will explain. But why did we become interested in air pollution in the first place for Parkinson's? Well, because we found that part of these particles that are called 2.5, that's a micron size, a large part of these micron-sized particles actually contain nano-sized particles. So nano-size are really nanoparticles. And where do they go? They go everywhere. And these mouse studies showed us that nano-sized particles also end up not only in the lung and olfactory bulb, so your smell can be affected, but also in the brain, okay? And microglia and neurons can be affected by these particles. And that has been shown in animal models quite a bit. So why do we go to Denmark? Well, because, and Denmark is, by the way, down here. This is the European map. And you can see Denmark here. Denmark has 2 systems. One is an air monitoring system and modeling system that is nationwide. The other system is a healthcare system that records all ICD codes nationwide and has done so since 1977. So you can go back and find every Parkinson's diagnosis ever in Denmark, and you can link it to air monitoring data. And why can you link it to air monitoring data? Because they also have a population registry and they know where people live. So we can find out where people lived, and we can find out what the air pollution was at their home doorstep. And we can find out who had Parkinson's. And that's exactly what we did. And you can see here we had almost 1,700 cases enrolled. These were all cases between 1995 and 2009, diagnosed in Denmark for the first time. And we collected from their population registry 1,800 controls. And we estimated traffic-related air pollution using nitrogen oxide, but this is really just a marker of the mixture that comes out of the tailpipe. And you can see what comes out of the tailpipe of cars here. It's kind of a fog, and in that fog are particles and gases. NO2 is just a tracer gas. It doesn't mean that it's NO2 that gives you Parkinson's. It means that something that comes out of the tailpipe probably does. And what did we find? A 16% increase in risk per standard deviation increase in this mixture. And, you know, depending on whether you're at the low end of the exposure or the high end, your risk goes up to 2-fold or more, as you can see here, with every increase in this exposure. And this is the case in Copenhagen and suburbs. It's also the case in provincial cities, not in the countryside, probably because we don't have as much pollution as we have in the cities. And in addition, we were able to also look at the genetics of these individuals. And again, it's a gene-environment interaction story. If you are genetically prone to inflammatory reactions, that's when your risk is almost 3-fold, when you're exposed to high traffic pollution. And it actually kind of confirms what we saw in animal models, where you had to expose the animal and you saw that it was not just the neuron that reacted and got poisoned, it was also the glia, which is actually an immune cell, that then started reacting to what was happening and generated inflammation. So it's an inflammatory response probably in the brain, in the periphery, that also contributes. So we definitely need public policies to reduce air pollution and pesticides. And yeah, I'm not talking, I guess, about lead this time, but this is the group who worked with me, Dr. Bronstein and his fellows, Yvette Bordelon and Adrian Keener, Dr. Kim Paul, who has done 10 years of studies with me, Dr. Custers, who is a genetics genomics person, my colleagues in genetics, Dr. Horvath and Sinsheimer, and these are students who worked with me seeing all these patients in the early 2000s, and this is the more recent crop. You can see it needs a lot of manpower to do these kind of studies, and without my students, we wouldn't have done any of it. Thank you so much. And of course, I cannot stop by not thanking our funders, NIH, the Environment Institute, the Department of Defense, Michael J. Fox, the Parkinsons Alliance, the APDA, and many, many donors to our program, including family members and patients.
Terrific.
Thank you very much.
Yeah, you're welcome. Thank you very much. I like that last slide. It's nice to put faces to the names in your presentation. Fascinating how much data you've collected, fascinating in how you've collected what with those school top sensors and all. And we have a lot of terrific questions flowing in already from our attendees. So Monique and I will alternate in lobbing some of those your direction. I will start, I think, with the most recent one because, well, no, there's been more since then. So I'll start with one from Robin, actually, because metabolizing was a key section of your presentation. Is there a way to tell if one is a slow or fast metabolizer?
Yes, indeed, you can actually get yourself genotyped for these polymorphisms, these variants. Mm-hmm. However, I would be careful to recommend this. I mean, you can go two ways. You can find out whether you're sensitive and susceptible, but you can also try to avoid exposure. And by finding out that you're sensitive to organophosphates does not tell you anything whether you're also sensitive to neonicotinoids, or by finding out you're sensitive to organophosphates using a different kind of pesticide. If you're replacing one agent with another, you don't know whether you might not be sensitive to that one as well, or you might be, sensitive to all pesticides. So, but yes, you could actually find out whether specifically for these organophosphates you are sensitive or not.
And sorry, maybe I missed it, but how would we do that?
Well, you would have to go to a genetics lab and find out whether, PON1 gene has these variants, your own PON1 gene. Mm-hmm.
Got it. And do you think the genetic identifiers in the commercial market out there will identify whether one is a slow or fast metabolizer?
It depends on whether they genotype these two variants. I'm not sure that they cover it.
Mm-hmm. Okay.
We specifically genotyped because we were, we knew that they had this function, and yeah, we specifically went after those.
Mm-hmm. Okay. You talked a little bit about how, Monique, why don't you give it a go? I see you're back unmuted.
Oh yeah, I'm here. Well, so that's interesting about the genetic testing. We put a poll in the chat. I don't know if you saw that, but it looks like 70%, was that right, of people, was that right, that had already had a genetic test?
Yeah, that's what I said.
And I'm sort of curious about that, and whether if people who had the genetic test actually had tested positive for any of the genes. And do you know how many genes there are that people can actually test for right now?
Well, this is the tricky part. So when we talk genes, we talk two different types of genes. One are the disease genes, and the other are the what I call gene-environment interaction genes. They're actually not disease genes. None of these, PON1 doesn't give you Parkinson's. PON1, however, detoxifies agents like organophosphates. It also does other stuff. That's why we have it, because we weren't created to defend ourselves against organophosphates. But it has other functions. This enzyme has other functions as well. But basically, what we are challenging with environmental exposures are our detoxification systems of cells. And that's not what is being tested at all. What you're being tested for are the genetic variants that in families increase risk of getting Parkinson's, not the variants that may keep you healthy if you're exposed to a toxin.
That's helpful. Thank you for clarifying that. And you presented some results on a meta-analysis, I think, where you had different rates from different studies, and then you presented sort of an overall odds ratio or risk. One of the questions in the chat was about, incidence in other countries, and what do we know about Parkinson's rates in other countries, and how do they correlate with pesticide use in different countries? Like I know, for example, China has really high rates of pesticides. Do they have higher rates of Parkinson's, for example?
Yes, that's a really great question. And we always run into this problem of, does a Parkinson's registry exists? Because how do we count Parkinson's cases, right? In Denmark, we can. And in Denmark, there is not at least from 1995 through 2009, there wasn't an increase in the incidence of Parkinson's. But, you could say, what does that tell us? Well, not much about pesticides, because it's not a country that uses a lot of pesticides in its agriculture. And definitely air pollution went down. So if anything, it has become a little cleaner over that time period. Other countries like Holland, yes, they have done studies of pesticides, and they also find links to Parkinson's disease. That's a good one. France has done studies in farmer insurance systems, and my colleagues there have also found links to Parkinson's disease. Countries like India, you don't even count death. They don't have death certificates. They certainly don't have the system to systematically count Parkinson's cases. And that's unfortunately the case for most developing countries that are drowning in pesticides. They don't have a system that counts cases. So we are very fortunate in California to have now a system that counts cases. We have a Parkinson's registry. It's not fully functioning yet. I'm just trying to get access to it actually for the first time. We had to simulate a Parkinson's registry for the studies I've done so far, and that is really expensive and really difficult and takes a long time. But California now has a Parkinson's registry. I hope we keep it alive and fund it. And the rest of the US doesn't. And the rest of the US also doesn't have a pesticide use report system. So they couldn't do these kind of studies.
Fascinating. It's diabolical in really the way we can't get that data to overlay the rest of the pesticide data.
I'm going to let you answer the next question, but I am going to post in the chat really quick for anybody who's interested some information on the registry that we have in California.
Yeah, good. Great. And the PANC.
I just see that somebody said Nebraska has a registry. Yes, they do, but it's a volunteer registry. I don't trust it. Anything that's volunteer, certain people volunteer to be registered, others don't. It's not systematic. If it's not systematic, we can't do science with it, unfortunately.
Interesting. Okay, next question comes from Christopher, and it echoes, you started to talk about food being one of the sort of vehicles by which we in urban communities like the Bay Area where we live might take in pesticides or toxic chemicals. What about the classic argument of organic versus non-organic and rinsing and washing your food before you eat it? Tell us a little bit more about that.
Yeah, absolutely. Very good question. So first of all, there are some pesticides that are called organic pesticides. Don't trust it, because the number one organic pesticide is rotenone, that because it's from a tree root, and it is being used to give animals Parkinsonian features. It is one of the pesticides that definitely kills dopamine neurons or impairs them. And there are organic farmers who have used rotenone, who actually both the farmer and his wife came down with Parkinson's. I'm not saying that's the reason, but there are anecdotal stories like that. So what is a pesticide is not just what chemistry gives us, it's also what's, of course, in certain plants, because that's how plants defend themselves against insects from being eaten. And so organic pesticides, be careful. The other is, yes, I have a colleague who actually studied this, what is on food and fruits and vegetables. And he did that by giving 2-year-old children, or mothers of 2-year-old children, organic fruits and vegetables for 2 months. And he measured the pesticides in the urine of the kids. And he found chlorpyrifos and all these other pesticides in the urine of the kids prior to changing them to an organic diet. And 2 months later, hoping the mothers fed them the fruits and vegetables he provided with his service, he actually found that the levels of pesticides had gone down or weren't there anymore. So these kids, you know, being fed the organic products really got rid of the pesticides. But it was very expensive, and, you know, we know organic fruits and vegetables are more expensive than others, and many people can't afford them.
Mhm.
Well,
It's still a good idea to rinse our food almost regardless of
Yes,
whether organic
or
or
peel
not.
it. Yeah, unfortunately, peel it. And that also gets rid, of course, of lots of good vitamins that are in the peels. When you peel apples, you know, you lose a lot. But on the
Right.
other hand, you get rid of pesticides.
Sure.
So speaking of pesticides and agricultural pesticides, there's a couple of questions that have come in through the chat about people who are concerned about farm workers. Obviously,
Yes.
they're very exposed to pesticides, and so are you? I guess couple questions. One, have you seen an actual increased risk in farm workers and their families? And what are we doing to protect the farm workers? Is there any outreach to these communities to sort of educate them about the risks? And, do you make any public presentations to these communities, and are you sort of involved at all in that?
Yeah, so first of all, this is, of course, a disease of older individuals, and farm workers are younger. And of course, we have our Mexican immigrants who stay in California. But when I started my studies in the Central Valley, most of the Hispanics were less than 50 years of age. And a very small proportion were already at the age where you would see Parkinson's disease, which is over 50. They, however, have been aging into that age range, and we have enrolled about 160 Hispanics now who have Parkinson's, and I'm very proud of that. The problem there is our healthcare system. I hear from colleagues who work as neurologists or general practitioners in the Central Valley who say, I clearly see people walking around Parkinsonian, and I know they're not getting treated because they're not seeing specialists. Even their primary care physician may or may not recognize it. They never get diagnosed. We actually did see that also in our study. All of our Hispanics on average got diagnosed 3 years later than the non-Hispanics, and they were worse off by the time they were diagnosed. Now, we have actually, one of my former students who started the study with me in 2000, got her PhD, then went to medical school, went back to be a physician in the Central Valley, and now is coming back to do research. She's also Hispanic. And she just got an NIH supplement for my study to reach out to the Hispanic community in the Central Valley. And we're very proud of that. But it's been a long battle to convince NIH to fund something like this. I'm really happy. And Michael J. Fox currently is also helping a postdoc of mine who is also very interested in the Hispanic community. She herself is Hispanic. And so we have now 2 individuals in my group that will concentrate on this population, but it's not an easy one to reach. You basically really have to have funding for a lot of outreach efforts.
So it sounds like one of the issues is just an underdiagnosing.
Yes.
And so there's a disparity going on with underdiagnosing.
There's a disparity in underdiagnosing, in undertreating. And in the former years, I also heard that elderly who become disabled may actually choose to go back to Mexico because the treatments are cheaper.
I know that we as a community, the Parkinson's, the pediatric community is just starting to really do some outreach work and to try to increase our efforts at diversity and inclusion within the Parkinson's community.
Right.
That's right. The disparity group that we talked about in the intro. I have a question here. I'll lob you, Dr. Ritz, from Margaret. Do you know, this is rather specific about DDT, and your presentation did not cover DDT, so maybe you'll choose to pass on it, but I'll just lob you the question. Specifically, do you know how childhood exposure to DDT, like mosquito spray, affects one's neurological system?
There have been 1 or 2 studies of DDT and Parkinson's. One was actually with serum samples in Finland that were collected in the 1960s. And going back, so Finland also has a national health system, so they were able to find Parkinson's patients, and then they were able to go back, and a number of them had participated in a Finnish big study where they had given blood samples that were stored over a very long period. So they could go back, and there were suggestions of an increase in risk, but it was not statistically significant. But that's one of the very few studies that ever looked at this question, because it is so hard to look at. Because you basically need these serum samples from 30, 40, 50 years ago to find what, who was exposed and at what level. And to make people remember what they were exposed to in their childhood, some people may remember, others just don't, or didn't know that it was DDT. So it's very hard to study.
And when you talked about the case control study, the PEG study that you did, and my understanding is that's how you gathered that information. These are people who are newly diagnosed with Parkinson's, and then you're sort of retrospectively asking them about their exposure history. Is that right?
Actually, not their exposure history, but their residential and occupational history, because we have the Parkinson's use report system to match up their addresses with exposure records. So nobody remembered anything. The household use, yes, they remembered, yeah, we used this kind of RAID, we always had this, and we used it in this way. But for the other, where we had all the specifics on pesticides, we actually used the pesticide use reporting system and overlaid it with the maps of the homes and the workplaces. So nobody had to remember. What they had to remember is an address.
Got it. I think that's a common question for people who get newly diagnosed. When they hear about the pesticides exposure, they wonder, what was I exposed to and how was I exposed? I mean, there are 2 questions in the chat specifically. One from Michelle, who's asking about growing up in the UK with a 200-year-old home with lead pipes and wondering if that could have potentially caused her PD. And then another question from Karen Garrison about parathion and its relationship to Parkinson's. I think she said she was exposed working as on a farm as a teenager. And I think that's a very common thing for people to sort of ruminate on, what was I exposed to? So do you have any comments about the parathion or the lead?
Well, it's possible. It's definitely possible. So the way I usually explain this is we know what Parkinson's is because we know what cell dies in the brain. It's the dopamine neuron. And the dopamine neurons also die over the aging process, right? The older we get, these cells, they are actually a cell population that until now, nobody thinks in neurosciences ever regenerates. It's like our ovaries. We are born with a certain number of these dopamine neurons and they don't regenerate. Other neurons have been found to regenerate over the lifetime. And so these neurons sit there and they just age. And then they're also working really hard to make dopamine. Dopamine is very toxic to the cell. The cell has to do all sorts of things to protect itself from the dopamine itself. And so over time, these mechanisms kind of start to fail, and then certain cells start dying. And there is a certain rate of cell deaths. And actually, some people think that if we live 150 years, everybody would have Parkinson's, because, you know, just by natural aging, we would get there. But why then do some people develop Parkinson's in their 30s, 40s, 50s, 60s, and others live 100 years and, you know, are fine? Well, you could imagine that you're either born with not enough dopamine neurons. So if you hit that 60% of cells dead, so that the 40% still alive can't provide enough dopamine anymore, when is that in your lifetime? If you're born with not enough neurons, that may come earlier. So what I'm really also interested in is prenatal development. I want to know who those babies are that are born with not enough dopamine neurons, but there's no way for me right now to tell. The other is you're born with a good number, with the actual sufficient number of dopamine neurons, but then you have a hit in your childhood and you maybe lose, you know, 0.1%. And then you have another hit with another agent and you lose 1%. And then you can add up and you see how the curve starts to merge with that, you know, 60% dead dopamine neurons much earlier in life. So basically what I'm saying is it's a cumulative risk over a lifetime. And yes, there might be a lot of different factors that contribute.
Is there a way to test for how many dopamine neurons you have? Like, say you're 30 years old and you want, I mean, your father has Parkinson's, and you want to say, well, how at risk am I on based on my sort of neuron dopamine capacity or something?
Right. I would have that, because I would use it in my studies and, you know, and tell people to be really careful. Everybody wants to know this. Mm-hmm. There isn't, because, and the reason why there isn't, there are neuroimaging potentials. We can image how dopamine neurons work. But interestingly, you do not see a deficit until 1 or 2 years prior to being diagnosed. However, your cells are already, you know, at times when it seems like they're working perfectly fine, 100%, it's 45% of your neurons doing double jobs. Mm-hmm. Basically, you know, running faster, working harder. And therefore, the functional readout in the imaging, in the dopamine imaging, tells you everything's normal. Well, because every neuron that's still alive is doing double work. Mm-hmm. You know, so, and there's no way to count the neurons while you're alive. You can do this, you know, in brains that you collect after post-mortem. You can count the dopamine neurons, but you can't, unfortunately, there's no way to count dopamine neurons. There's only ways to see their function. And as I said, the function is deceptive. Until it breaks down.
Until it breaks down, right?
Yeah.
Here's an interesting question from Renee, and Renee may have read Ending Parkinson's book, but she says she lived and worked in a Navy camp, the Navy camp Lejeune in North Carolina in the 70s for over a year, exposed to the toxic water there from dry cleaning companies, etc. Virginia has declared PD and several cancers as presumptive diagnoses from the exposure. Would a genetic testing help in her case?
I don't think it does. The reason is these solvents, maybe metal exposures on the base, they happened a long time ago. Whatever it was, you probably can't trace it back to that time. But we do know that, you know, your dopamine neurons don't like metals, and they don't like solvents either. They don't like pesticides. And if you have enough of it, you know, some of these cells might just die an earlier death. And you might be genetically at a disadvantage, and therefore your dopamine neurons were stressed even more. But that doesn't really help you answer your questions.
Okay.
Here's a question from Janice about working in organic chemistry labs. Has there been any studies on people working in those labs and the risk of Parkinson's?
That's a really great question. And no chemistry lab studies exist, but there are anecdotal stories about chemists who, had intoxications by accident. Actually, one chemist, I think, also produced MPTP, and he got Parkinson's, just like these heroin addicts. And my own mentor at UCLA, he is a chemist, toxicologist, and he would always joke with me in the 1990s saying, Beate, you and your loss of sense of smell, every chemist loses their sense of smell because, they get hit by this and that and something else. And I've lost my sense of smell because of that a long time ago. Well, he was diagnosed with Parkinson's about 10 years ago. So, it's anecdotal. We don't know. It's certainly one of the possibilities. Yeah.
Wow. Back to the household. What warning signs should we be looking for on any household products that we worry might be problematic?
Good question. I mean, we mostly investigated pesticides. I would say keep those out of the house. Definitely not under the sink.
Right.
Otherwise, I sometimes, I actually use an air cleaner now when we have wildfires. I did move away from traffic. I would suggest that if you live close to traffic and you can't move away, use air filters. They actually work. They take particles out of the air. We have colleagues who showed that they really reduce particle load indoors. And remove chemicals you don't need if you can. Gasoline, solvents, remove them from your home.
Okay.
And I know that the Environmental Working Group does put out a lot of data on, pesticides. And one attendee is asking if you trust the data from that group, the Environmental Working Group.
Yeah, there's also the Pesticide Action Network that puts out really good information. Definitely. Okay,
great. And then, so we talked a lot about exposures, past exposures, and a lot of people who are joining us on the call today, already have Parkinson's. So maybe they're not as worried about sort of, preventing exposures if they already have Parkinson's. So do you have any recommendations for people, who already have Parkinson's?
Absolutely. I would say still avoid certain exposures, especially the ones that increase inflammatory reactions, because we know that's not good for the brain. So air pollution is one of them. Also, antioxidants are really good. So, healthy diet, Mediterranean diet is probably what your grandmother always told you, eat your spinach, eat your vegetables and fruits. And physical activity. We really saw, we asked patients about physical activity and controls as well. And we saw that physical activity was definitely really important to stay in shape and to not progress.
Do you recommend that people take anti-inflammatories? Another person is asking.
Yeah, that was a hypothesis a long time ago. It's not really holding up. I think it's too late in the process. When you already are diagnosed, you probably needed to really do that when you were in your 40s or 50s. When you're already diagnosed with Parkinson's, they're probably not helping you, and they have a lot of side effects. They cause gastrointestinal bleeding, and you don't want to be on anti-inflammatories unless your doctor tells you.
Right. I think that would probably be for the NSAIDs, right? But then there are also natural anti-inflammatories like the curcumin, the turmeric, things like that.
Right. That is fine. Yeah, as much as you like them.
Yeah,
definitely. Yeah. Alcohol is a good question. We don't know. There are studies that show that Parkinson's patients drink less alcohol, just like there are studies, that say they smoke less. And I would not recommend drinking alcohol or smoking to protect you.
Mhm.
But it doesn't seem to also be a huge issue in terms of increasing risk for Parkinson's. As with everything, do it in moderation.
Well, I've always been very interested by those results that show that there's sort of lower rates of Parkinson's in people who smoke cigarettes. And also, I think caffeine, right, intake is also.
Yeah.
I mean, is there anything that you would like to
Say
share
about that?
those?
Yeah. Yeah, I'm one of the controversial people on this because I say it's reverse causation. Even so, I have lots of colleagues who swear there is something good in coffee and there's something good in nicotine or whatever else there is in cigarette smoke for the brain. I don't believe it yet. The reason why I don't believe it is that basically my hypothesis is you lose your brain's addictiveness to nicotine once the dopamine system doesn't work anymore, because dopamine kind of is in this reward cycling loop, right? And so people who then want to stop smoking have an easier time stopping when they are already having a dopamine system that's not as responsive anymore. So they stop smoking, they have no problems. While the rest of the people whose dopamine system is working, they have a really hard time stopping smoking. There are, however, also the people who never pick up smoking. And we could argue, what is that? Why do Parkinson's patients pick up smoking at a lower rate? Well, maybe their dopamine system is already from birth not as efficient, so they're not getting as addicted. The same could be for alcohol and caffeine. That's not as addictive, but we do know that there's sleep disturbances. We know there's gastrointestinal problems. And we do also know that Parkinson's probably starts somewhere in the periphery before it is in the brain, or before you at least diagnose it in the brain. And people may stop drinking coffee because they have gastrointestinal symptoms due to their Parkinson's. They can't sleep due to their Parkinson's. And one thing they do is not drink caffeine. So to me, the jury is out on whether it's reverse causation, meaning once your dopamine system and everything related to Parkinson's that's non-motor feature is out of whack, you may actually stop doing things rather than these agents protecting you from Parkinson's.
Interesting.
But as I said, my opinion is controversial. Some people believe there is somewhere the holy grail substance in coffee or in cigarette smoke that protects your brain. I don't believe it.
And some of those studies that talk about caffeine being one of those ingredients in coffee talk about a second ingredient. Do you know what that one is? I can't remember. A second ingredient in coffee that can be helpful.
Coffee also has a lot of antioxidants in it, and I think some of the idea was behind coffee being protective that it's an antioxidant.
Okay. A question on the EPA. It's shocking to me, at least, is it to you, how unwilling they seem to be to take action against some of these terrible toxins and pesticides and solvents in the face of so much compelling evidence. Is this a political problem, or is it a money problem, or are we just not yelling loudly?
Right. I think it's an economic problem. There's such a huge industry that's selling this stuff. I mean, the egg industry needs it, or has been made to need pesticides to grow crops. We are made to believe that the only way to feed the population is through widespread use of these pesticides. Yeah, I think it's very hard to disentangle this now from our food supply and from how we live our lives. And terrible. EPA is very responsive to industry, even so it's the regulatory agency that should be watching, but they are very responsive to industry.
So for those listeners out there who have not yet read Ending Parkinson's. The EPA has not yet banned paraquat in the face of all this compelling evidence, or TCE, which is something we didn't talk too much about today.
Right.
And a handful of others, but those are the two glaring examples.
And I think that Todd and I, we did put together a list of action items that people could do. We can share with everybody later, phone numbers that you can call, organizations that you can get involved with, if you're interested in sort of fighting the good fight in that regard. And someone did ask in the chat, Dr. Ritz, sort of, did you share your work with other, public health organizations or legislator, or not to get political, but do you do any advocacy work yourself?
Well, as a scientist, you're always told you shouldn't be an advocate or else you're not credible anymore. But I'm actually also a public health person, and public health always gets involved in something. So I sit on expert groups, expert working groups as a scientist, and I, promote my opinions there and my research there. I sit on the Air Toxics Board as an expert, as a scientific advisor for California. I've been sitting on EPA boards when they were still using people like me who actually do research. I open my mouth when I'm asked by Parkinson's foundations and, certainly towards NIH. I do what I can, but I think in the end, I can't be the ultimate advocate because then I become non-credible as a scientist.
Mm-hmm.
Right, conflicts of interest potentially.
Yeah.
Let's see. Karen asks, how can we get registered with the different registries? So the quick answer is Monique pasted into the chat window the PANC, or Parkinson's Association of Northern California's registry site. That's one. Do you have other links that we can share with folks or sites that we can point them to, Dr. Ritz?
No, I think actually, if you really want to help the Parkinson's registry, you need to encourage your healthcare providers to participate, because the biggest pushback we got, and I mean, we got the law enacted in 2004. That was Arnold Schwarzenegger who signed it, and he was a good friend of Michael J. Fox, and I think it helped. So, but that became an unfunded mandate until only 3 years ago, meaning nobody wanted to pay for a registry. So we didn't have a registry, even so we had a law for a registry, until the state of California finally put some money behind it. And then it was curtailed to a great degree because the California Medical Association, and I don't want to blackmail my colleagues, I really, I need them, I hold them in high esteem, but they were very worried about the workload that would actually generate for their offices. And that's not a minimal worry, because I know how overrun some of these specialty offices are, and primary healthcare facilities are. They're trying their best. And then when the state comes and makes another mandate, reporting mandate, then they say, and who's going to pay for that? Right? And then I can't see patients if I have to report. Well, to the rescue comes electronic medical records. And I think we can actually solve this problem if more and more healthcare providers, first of all, are positive about registries and how they are used, and that they're really used to the advantage of their patients, not to their disadvantage. And the other thing that, they allow us to, they first of all buy into electronic medical records, and then that we are allowed to use the electronic medical records to populate a registry. That's the best way I see forward from, all this discussion on registries. So and we need a positive attitude from the healthcare providers, and not, we don't want registries.
So I think as patients, what we can do is when we go to our doctors, we can ask them about the registry. We
right.
can just
we can say, I hope you participate and in the Parkinson's registry, because I know they do some science that may help me in the end.
Right. I think Kaiser participates in the registry.
Yes.
We go to the
And the
Doctors-
UC system, yeah. Actually, I'm part of a Michael J. Fox funded validation effort and a big effort in the whole UC system to populate the registry in a good way. And that can be then expanded to other registries like Kaiser and other healthcare systems. And lots of doctors now are not independent anymore. They actually belong to a healthcare system.
Mhm.
So they also have to advocate within the healthcare system and not oppose these kind of movements.
So I think this is really important and worth reiterating. The registry data of who has Parkinson's and where you live and what your symptoms are and where you grew up is kind of the missing holy grail that's missing to overlay the rest of all this terrific research and data that you collected in your different studies. So the short answer is to Karen and anybody else interested, ask your healthcare provider, ask your GP, where is the registry that I can get involved in? Which ones are important? How can I do it? What websites do I go to? And it's not just, I hope it's not just Karen curious about that. The more of us that can go out and get registered, the quicker we can get this data loaded in for researchers and doctors like Dr. Ritz to lay over the rest of the toxin data they've been collecting.
And there is a really good good clip that I put in the chat. The Parkinson's Association in Northern California works a lot on advocating and doing work with the registry. And there's a little video, a recap of their conference last year, which was all about the registry. So if you're interested in learning more about it.
Yeah, the registry is also really a great tool to improve health services. So it helps me do my research, but it also will, eventually it will improve health services. So it's really important.
Yeah. We're nearing the end of our 2-hour window. It's 12:15. We're nearing the end of our questions, which is convenient. I will throw this up and in response to Michelle's question, what is the title of the book you referred to initially, Ending PD? This is probably showing to you as backwards. So I will tell you that it's called Ending Parkinson's Disease, a Prescription for Action by 4 different doctors and researchers around the world. It's fascinating. It has a very lengthy and detailed section to the last third of the book on preventing, advocating, caring, and treatment. So PACT is how they've abbreviated it, PACT. We will share with you a little bit of that. In fact, maybe we'll break away, Monique, unless you see any other questions that we'd like the doctor to field. You see any questions?
I just have one more thing I want to
Sure.
say. I actually, because of this talk, I looked online at, what is the funding situation for environmental research in Parkinson's.
Oh, good.
And I found that I'm the most funded person in the country on environment and Parkinson's, but that's very sad because I have much less funding than many of my colleagues for other diseases. And I think that really has to change. And my young colleagues will study other diseases that, get more funding. We need to get the NIH to take environment as a big E. And not a tiny E to be ignored.
And how do we do that, Dr. Ritz? What can we do as lay people to help in that effort?
So if anybody has, some political pull, knows people who do, write to Congress, write to the Senate, say we need Parkinson's research that's not just basic science. We also need research that looks at the environment. And I know that the autism speak groups did that very effectively, and they've got hundreds of millions of dollars invested from the National Institute of Environmental Health Science in autism environment research. We don't have that in Parkinson's anymore.
Interesting.
I wonder if that has to do with the fact that the populations, with autism, it's often children, and parents are a lot more politically motivated, perhaps, than people who are older.
Absolutely, absolutely. Actually, when I started my research, there was no funding for environment in Parkinson's, and I almost gave up. And then there was a young activist Parkinson's group that went to Congress and got some money out of them for environmental research. That's why I'm doing this. And I kept continuing. I'm very hard-headed, so I never went away. But my junior colleagues have no chance of getting funded. They have to establish their careers, and there is no more voice out there currently in Parkinson's that asks about this kind of research, environmental research. Unfortunately, that group of young activist Parkinson's patients died out. I don't know why. One of them was a lawyer who had severe Parkinson's, and maybe it was just that she was too affected. And, you know, nobody took her place. So we need activists for this cause.
There is a group now called PD Avengers. I'm not sure if you're familiar with them, but they are pretty strong activist group advocating for a lot of these issues. PD Avengers, if anybody is interested.
I think we have that link, Monique, if you have a second to paste that into chat.
Okay.
Meanwhile, I'm going to jump over and share the action steps that we talked about. I'm sharing my screen now, if you can see that. This is what we tried to keep this down to just a one-pager. It happened to grow over into a second page. We will send this out to all attendees. This is a small subset of the portion of the book, Ending Parkinson's Disease, that I referred to a moment ago. The last third of the book that is PACT. So preventing, advocating, caring, and treating. We use the first two, which are prevention and advocacy, to present to you, and you can learn the rest about the other two in the book. These have very specific contact options for example, the EPA administrator today is Michael Reagan, a direct line, including an email address, U.S. House Committee on Energy and Commerce, et cetera, et cetera. So please give this a read when we send it out. If you're intrigued by it, then I again point you to the book itself and the lengthier content that's in that. But other than that, I think that's all we have. Any other questions, Monique, that you think we can solve for before we log off?
Sorry, there was one other couple quick questions in the chat. If we could try to answer them, I think that would be great. I'm not sure I understand completely, but Eileen is asking if decades of use of prescription dopamine reuptake inhibitors could be related to developing Parkinson's.
That I know there are some hypotheses out there. So far, that has not been shown.
Okay, great. There's another question from David about, is there a connection between Parkinson's tremors and the same kind of tremors you get when you are cold or nervous?
That's a good question. No, I think that's not the case. Those are different mechanisms. There's also something called essential tremor. It's also from a different mechanism, brain mechanism.
And Ellie asks what research you're working on now, Dr. Ritz.
Yeah, so currently I have a small study funded for depression in Parkinson's because I think it's a really important topic. And the other one is on the microbiome. So we are really interested in seeing how the microbiome contributes and whether there's something we can do to improve what the microbiome does, because it's this big detoxifying organism inside us that we know very little about. So those are the two areas that we're currently studying.
Well, that actually brings up a question I had before when you were talking about one of your studies on pesticides in the biome. Can you elaborate on that connection there? Is the idea that you're ingesting pesticides and the pesticides are in the gut biome? And then
yes, so it's twofold. There are actually microbicides that are used out there as pesticides, or fungicides. So these microbicides may also, when you ingest them, change your gut microbiome. And the other is microbials. There are also certain microbes that are being put out there. And how do these microbials then influence your gut microbiome? And then third, how do pesticides, when they enter through food or enter through the gut, more or less, how does the microbiome then act in order to either activate them and make them more toxic or detoxify them? So those are the kind of questions we are trying to answer. It's very complex and it's not easy, and you know, patients also have hesitation sharing fecal samples. It's not that easy, but you know, I really commend anyone who, you know, is volunteering, and it's in the Central Valley. It's the same group. You know, we hope we get enough people.
Good. And last couple here, back to the question on advocacy. Christopher asked, don't we have APDA, Michael J. Fox, et cetera, doing advocacy on our behalf out there also? We know that.
Yes, definitely you do, but it depends on what kind of advocacy we are talking about. They are not the ones going necessarily to NIH to ask for more certain types of research. They, you know, they have their specific interests. APDA is more a healthcare related advocacy group. Michael J. Fox has discovered the environment. So they are now funding me, but for the first time last year. You know, they had not funded me in the last 20 years.
Wow. And they do a lot of research on cause and treatment,
Yes.
not so much
Yeah,
environmental.
and genetics dominated a lot of it for a very long time.
We'll make this nice question our last one by Margaret asking, is Dr. Ritz able to accept private donations for her pig study?
Yes, I am, but it has to come as a check to me for UCLA. So it goes into a UCLA gift fund. So you can send it to me, my address at UCLA, which is, yeah, Department of Epidemiology, UCLA School of Public Health. You find the address online, and you can say it's specifically for Dr. Ritz's Parkinson's research, meaning I can't do anything else with it, and I won't. And it goes into a gift fund that the university administers, but I get all the money.
I will try to put that in the summary that I send out in an email to all attendees in the next day or two so that we can make that available to them.
And by the way, a gift started me. I would not be doing this unless there hadn't been a luncheon that I was the junior researcher in 1996, I think, presenting my ideas. It was just ideas at the time, you know, using that pesticide registry, etc. And one of the people at this luncheon said, I'll write this lady a check. And, you know, it was Christmas. And the department got this check and said, Beate, you got money. and I used it to do a pilot study. And that was the pilot study that I needed to get the big NIH grant of $2 million.
Wow.
It's amazing how it sometimes works. So thank you to that anonymous donor.
Great. Well, now we're getting flooded by thank yous in the chat window, so I will not read all of those, but Monique and I will echo those folks and say thank you very much, Dr. Ritz, again for sharing your time on a weekend, no less. It's fascinating all the work that you're doing. We hope you can get more funding and more data to overlay all of that toxin research and data that you've got. But on behalf of all of PD-Active, thank you very much for your time today.
Thank you, Todd. Thank you, Monique. And thank you all of the audience who I couldn't see.
Thank you.
All right. And attendees, thanks very much for joining us today. We appreciate you being among us. And again, if you have a minute to go to the pdactive.org website and make a donation for the talk, that would be terrific. Otherwise, we hope you enjoy the rest of your weekend. And thanks very much. Adios.
Bye-bye.
Good weekend.